So now we're going to return to our previously scheduled program, Enzyme Lab. All right, so who can, in a nutshell, describe the objective? What's the objective in a nutshell? But that's like that's like an idiomatic expression. It means like put it, just kind of summarize it briefly.
So we're looking at varying rates of substrate, the hydrogen peroxide, and how it affects the rate of reaction. We're looking at the O2 that's produced with the enzyme catalase.
Wait, so let's start one more time.
So we're looking at how varying levels of the substrate hydrogen peroxide reacts with the catalase enzyme. And we're measuring the rate of reaction via the oxygen levels.
Okay, and be a little more specific about the independent variable. What is our independent?
So we're looking at the substrate concentration. So we have different ratios.
So the concentration of the substrate.
Yeah.
I didn't know if I heard that totally. We want to be very, very precise, because that's how we are going to be able to kind of keep track of what we're looking at, how we analyze things. So we are going to vary substrate concentration. And we saw that last class. We just do that by making these different dilutions of hydrogen peroxide solution, and then keeping everything else equal, the same amount of enzyme, the same volume, the same procedure. Let's see how the reaction rate is affected. What is a compelling hypothesis for this lab? I know most people at least wrote it down.
So as the substrate concentration increases, the reaction rate will also increase. But then at a certain point, the enzymes will be fully saturated. So even when more substrate is added, the reaction rate won't increase anymore.
So exactly. So did everyone hear? Lily stated that in a really just very clearly, which is going to be important as we move forward. That really makes sense. So if we're looking at substrate concentration as our independent variable, right? And reality is it's going to be like 0. It's going to be kind of 1 to 4, 2 to 3, 3 to 2, 4 to 1, 5 to 5, like 100%. So we're doing 6 different runs of the experiment, right? We're doing it with no hydrogen peroxide and not 5 to 5. I was right, 5 to 0. This is like pure H2O2 solution. It's actually 3% solution. So pure 3%. So we're going from basically 0% to 100%. 100% of the 3%, I should say. We'll do the experiment 6 times. And in each case, we are going to be looking at the reaction rate. And according to Lily's hypothesis there, we're going to expect that it's going to be having some kind of a direct correlation up to a point, whereas you add higher substrate concentration, you're going to have a faster reaction rate. And at some point, if you saturate the enzyme, you'll get to some place, even if you add more hydrogen peroxide or increase the concentration, I should say, it's not going to get any faster. It's not going to slow down, but you'll be at some maximum rate. So this would be what we would expect, if what she's saying is supported. Exactly where this saturation point, who knows? And again, if we don't ever saturate, it might just keep getting higher and higher. Or if we're saturated from the beginning, it might just never get faster at all. How are we going to get the reaction rate, right? Substrate concentration we get just by making dilutions. How do we actually get the reaction rate for each of these trials?
On separate graphs for each trial, we have to calculate the slope.
Exactly. So this is going to be what I want to talk about now, where people get really confused. So I want to spend some time making sure everybody is feeling comfortable with how are we going to get the reaction rate. For each of these trials, we're going to need a value here. What is the value? Where does this dot go? Does it here? Does it here? Is it here? Where does the dot go? To get that value is going to be a process of making a different graph, a graph of time versus oxygen for that trial, right? This is trial one. Trial one. This is actually trial two. This up here is like trial 5. I think this one we call trial 6. Right, so each of these dots is going to be a process that we have to do with the data that we get from running the entire experiment, running the trial. You saw Tina and I last time, Mark, what's the little bubbles? Where's the level of the bubbles now? Right? Other things to remember before we kind of get into the actual data. Bubbles are coming up. Bubbles are bloop, 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 bloop. And as the bubbles are going up in this water-filled burette, they displace the water that's filling it. So if there is no oxygen in there yet, the water level is going to be at 50. So that's where we start. So time equals 0 with no oxygen. There's 50 mils of water in there. As the oxygen bubbles up, it's going to push the water down. Now the oxygen is coming in here. And now maybe the water level is down here. So let's say this was 42 mils is my level of the water. What does that tell me about how much oxygen is in the tube?
Eight mils of oxygen.
Exactly. How did you calculate that?
You just subtract 50.
Exactly. So the mils of O2 is just going to be 50 minus the mils of H2O that are left. So that's going to be important. When we look at the data that you have on your module is the direct reading off of the burette. So the burette reading is actually 50 minus that reading is going to give you how much oxygen is up here displacing the water. So that's going to be like when you get your data, the first thing you're going to have to do is convert it, because I don't care about the burette reading. I care about the amount of oxygen. So the first thing you're going to have to do is make sure you convert that into oxygen. And then, again, I will kind of review this. For each trial. You can do these on separate. It is possible to do them on one large graph in different colors, but you're basically going to do time in seconds. Etc. versus mils of oxygen. And you're just going to plot it. Again, this is just an example. And again, you are going to have dots because we're taking data at points that are just every 5 seconds. But you kind of, in your mind, you kind of try to smooth it out. What do you think it really looks like? Something like that. So let's talk about, first of all, making it really clear, this graph here even though even though this one now looks really ugly. Remember, even though this one also in an ideal case kind of goes up and over, they are so not equal, right? I really want to make it clear there's two fundamentally different graphs that you need to be doing in this lab analysis. This graph here, reaction rate versus substrate concentration, this is what we ultimately want. This is the thing that's going to allow us to check out if our hypothesis is supported or not, right? We are interested in substrate concentration versus reaction rate. So ultimately, what we want is to be able to make this graph. When we can see this graph, then we'll be able to answer the question that we stated in our objective, which is, what's the relationship between substrate concentration and reaction rate? However, to get the values of reaction rate, each of these dots here, whatever the reaction rate is in each of these trials for each of these substrate concentrations, right? Each trial is at a different substrate concentration. For each trial, you are going to have to do this. So this to calculate reaction rate, at this concentration. So let's say this is trial, I don't know, let's say this is trial 2. What you would do is you would plot this out. And again, remember, this is not substrate versus oxygen. I mean, substrate versus rate. This is time versus oxygen. So it's really different. This is a graph to calculate numbers so we can put it in our other graph that we really care about. In this case, we can see as time goes on, oxygen seems to be increasing because that makes sense. Hydrogen peroxide is breaking down, making more oxygen. What is happening here?
All the substrate's been used.
Exactly. Here, the substrate is all been used. There's no more hydrogen peroxide to break down, so no more oxygen is being made. So the amount of oxygen is just going to be, flatlined. There's no more increase in oxygen because we're not doing anything. So here, the reaction is over. So this is important. We don't care about this part of the graph. When we are going to calculate our reaction rates, we want the reaction rate when the reaction is running, right? That's the dots on our graph of reaction rate versus substrate concentration are going to be what is the rate when the reaction is really rolling along. So what we need to do is we need to make sure we calculate this value from when the reaction is strong, not when it's dead. So we go back over here and we say, holy moly, the reaction's dead. We're going to completely ignore that part. The sweet spot is going to be where we see it kind of going up in a kind of nice, linear kind of area here. So this is what we care about, right? We want this part. We don't want this part. Does that make sense? So this is important. A lot of people kind of just like draw a line, like, get a ruler and like draw a line, like, I don't know, from here to here. Why is it not working? Oh, interesting. I try using a metal ruler with my fancy stylus. It doesn't work, but plastic, I bet you will. Plastic does. So if I drew, oh, there's my best fit line from where it starts to where it's ending. It's like, nope, that's really bad. Because if you take a look, it is not a good approximation of what this thing is doing when I'm actually interested in that strong reaction. So I'm going to back up here. Boom. And instead, I'm going to draw my best fit line. Let's maybe like this. So my best fit line. You do your best. So it's kind of approximating what is the slope while this thing is really going strongly. It's not going to match up here. It's probably not going to match up down here either. Right. The beginning is when everything's mixing together. You're not going to really trust things at the very beginning. And in fact, we're going to see even more of the reasons not to trust it there. Does not have to go through zero. Right, so this requires a little bit of judgment to figure out what is the best fit line. Because you could imagine, yeah, that line looks good, or maybe that's a little better, or it's like you want to different. If you have 3 different people, you'll probably have 3 slightly different versions of the line, but they should all be pretty darn close. And then what do you do once you've drawn your best fit line?
Calculate the slope.
You're going to calculate the slope, right? Because that slope is going to be our quantitative measure of the reaction rate. This is how many moles of oxygen per second are being created in this particular trial. So we just have to go, let me like, I usually go like 10 away from each other. It makes it easier. Then we can look at so we have our delta Y over delta X. That is rise over run. And that slope, sometimes people call it M, the slope is going to be mils of oxygen per second. And this is going to be our measure of reaction rate. So let's say, I'm just going to make up some numbers here. So let's say on my graph here, it looks like this. And then I can say like, okay, my calculation for this slope, and right, I can pick any 2 points. If you pick any 2 points on the same line and do the rise over run, it'll be the exact same slope number. It doesn't matter which 2 points, pick ones that are easy. I am picking the one at 20, it's 10 here. And at 10 seconds, it's 5. So I can say delta Y 10 minus 5, right? 10 minus 5. My delta X is going to be 20 minus 10, which I designed to be 10. And this is going to be 5/10 is 0.5. So then I have here, okay, this is trial 2. I go back to my original plot here, which is so freaking so ugly. Let me go do a new one. Oops. So I have my graph I want to do. Again, I go back to this calculation. Again, let's say this is trial 2. Trial 2, which was 2 mils of hydrogen peroxide out of a total 5. So this is like 40% solution. So then I go back to this thing I just drew here. And I can say, aha, at 40%, let's say this is 10, 20, 30, 40, 50, 60, 70, 80, 90. I can say it's 0.5. So that's where I'm going to put my dot. So in order to get 1 dot on this, this is trial 2. What I need to do is I go get my time versus mils of oxygen, get a best fit line, calculate the slope, and then use that slope value as my reaction rate for that trial. And then I take that number, which I've gone and done all that, and I put it there as one dot. You know, you're going to have to do the same thing for trial 1 to get whatever dot fits here. The same thing for trial 3 to get whatever dot fits there. So is this making sense? Okay. I'm going to go share. I'm going to show a little more stuff to kind of help you out with this. So again, in the pre-lab module, I have a couple of little videos of people describing, you know, how to get reaction rates using a graph as well. And I've tried to write it up and I'm just talking about it here. Remember that since you're using this graph for quantitative analysis, you have to make a pretty graph. And it's got to be big enough that you can, don't make some dinky little thing, you know, make sure it's, you know, fills up a page or something so you can actually draw slopes and calculate things accurately. You know, again, we've done all of this part last class. I went through all of this. So here's the data. All right, so actually I've been playing with it already. Whoops. So if you take a look at the data, let me make it a little smaller. Let's make it a little bigger. How about 120%? So you've got the data and group 1, group 2, there's 5 groups. And this has the average readings for the burette. And then each trial is on its own tab. Trial 1, trial 2, trial 3, trial 4, trial 5, trial 6. Let's take a few moments and look at trial 6 because it's actually important. What would you expect for trial 6? What would you expect your results to look like? What is the substrate concentration in trial 6?
0.
It's 0. So what should we expect in terms of the reaction rate?
No reaction.
No reaction, right? In fact, this should actually be a good control for us, right? If there is no substrate, there better not be any bubbles. Otherwise, what we are measuring is not the reaction, right? So it's important to double check, like, all right, if I don't give any substrate at all, I better not see a reaction. This is like a test to make sure our system is working. Let's take a look what actually happens here. I'm going to create a new, let me increase the zoom just slightly so you can see it a little better. Yeah, let's go maybe to So if I want actually like, mils of oxygen, how am I going to do that?
Subtract it from
minus
50.
the buff rate.
Yeah, it's going to have to be 50 minus whatever the burette reading is. How do I do that? If I'm in Excel, how do I tell it I want a formula that says it's 50 minus whatever is in this cell here?
Would it be equals
Yeah, remember, it always starts with an equal sign.
and then 50 minus B3?
Yeah, it's going to be 50 minus G3 because there's the average one there. And if I hit enter, 50 minus 50 is 0. All right, that's good. And if I want the exact same thing for all the different time points, what do I do?
You just drag it down.
Yeah, I put my cursor right in that lower right-hand corner till it turns into that plus sign. Hit down the left mouse button, drag down, let go, and kazing. It just did it. So we're seeing something weird here. I don't see all zeros here. I'm going to do a quick and dirty little graph of time versus mils of oxygen, and let's see what happens. Yeah, I should probably, why? And my other axis, it's just seconds. So what do I see here?
a.m.
Anybody, just tell me what you see. And also
A reaction rate.
what's that?
It looks like an average reaction rate of, about 6, just under 6.
Wait, no, so reaction, how would you find reaction rate? You want to be careful about, remember this, our y-axis, this is just mils of oxygen, how much oxygen we've made. In fact, what is the reaction rate all along up here?
Constant.
It's constant. Let me see if I can add in. Kind of, if we add the lines into it, it looks kind of like this. So what is this saying? Up here, what's the slope of all of this?
0.
0, right? It's a horizontal line. So here, the slope is 0, which is what we expected, right? Slope of 0 means no reaction. But there's something going on in the beginning. The first 10 seconds is definitely not a slope of 0. So what is happening? This is why we do these kind of controls, because you want to, our assumption is we should just get a slope of 0, no reaction if there's no substrate. But here we see like, okay, no, something's happening. So what is happening?
Oh, good.
No, say it, Dana?
Oh, the catalase reacted with the distilled water.
So that might have happened, although that wouldn't make sense. And we also saw after 10 seconds, it's completely flat, nothing's happening.
Could there be contamination?
Right? So it could be contamination as well. But again, we look at this and say, it's just, there's something weird happening right in the beginning, but then it stabilizes. This is where it's worth going back and remembering the actual procedure, like why you actually looked at how we got the data and not just giving you data. So here, let me do this. This is going to be worthwhile. Boop-a-doop-a-doo. So here is the system. And let's spend a few moments and really think about this. All right, what happens when we first start the experiment? We have, what's in the syringe? Right as we're about to start a trial.
Water.
Yeah, so for this trial, what was in there? What was in there? It was, if you want to be exactly specific, what was in
Oh,
there?
sorry, the syringe, not the burette. We have our concentration of solution. We have the water and the hydrogen peroxide.
Or in the case of trial 6, it is actually pure water. And how much water or how much solution?
5 milliliters.
Yeah, there's 5 mils of whatever we are our substrate dilution that we are going to be using. So it's waiting in the wings. We have our enzyme solution sitting here, waiting to mix it in. And what happens at time equals 0? And we're going to start.
The oxygen from the rubber tubing goes into the burette. And then the water starts flowing. Could that be why?
Well, so where does this 5 mils of water go?
Goes into the catalase.
It goes down into here. So we've just added 5 mils of water or fluid into our flask here. What used to be in there that just got displaced by that 5 mils of water?
The air?
Yeah, 5 mils of air all of a sudden is like, whoa, I just got bumped out of my little comfortable home here in the flask. Where's it going to end up going?
You're at.
The burette, right? So now when you're thinking about it this way, and if we go back and look at our graph here, what is probably happening here? And again, this is, trial 6, pure water. So what is going on here?
So that's the oxygen that's in the burette from adding the water.
Yeah, I wouldn't even call it oxygen, right?
Just air.
Right, there was just, room air that was sitting in the flask that got displaced and went up the burette, which then displaced the water. So this first, 5-10 seconds here is just a big globby bubble of air that is leaving the flask and going up, that is due to just shoving 5 mils of our solution into the flask. Does that make sense? So here you can see this control, this doing this thing with distilled water. It's telling us a couple of important things. One thing that it's telling us is, it's telling us like, oh, don't trust the first 5 or 10 seconds because there's actually a big contaminating blob of air going up into the system. That is not due to the reaction. But then what's it telling us after 10 seconds?
That is pretty trustworthy data.
Yeah, after 10 seconds, then it really does seem like there is no bubbles happening just due to the water or due to the mechanics of the system. So it's telling us, don't trust the first 10 seconds, but it is okay. Afterwards, your assumption that pure water is not causing a reaction is actually accurate. Right? So, understanding why it's important to do a control like this, you get to actually, in this case, it's kind of showing us two things. It's showing us actually there is something to worry about. Some of our original assumptions were not true, but then in the bigger picture, nothing to worry about as long as you make sure you ignore the first 5 or 10 seconds. So any questions about that? Like, it's important to understand this before you do the next parts of the data analysis, because otherwise you might believe the data point at 5 seconds. And this is saying, like, don't believe the data point at 5 seconds.
Would this information change our best fit line?
Yes, of course. It means that you're not going to take that 5 second, that reading at 5 seconds into account when you're doing your best fit line.
Got it. Thank you.
Yeah, so for instance, let's do another one just to kind of give you a sense of how this goes. Your trial 3. I'm going to do mils of oxygen again. And again, how am I going to have it calculate mils of oxygen?
50 minus the
It's going to equal 50 minus, whatever this puppy is.
G3.
And then I can use, and this should be 0, but somebody zeroed it at 49 and didn't bother to change it. So whatever. All right, so here we have data, oxygen versus time. Let's, again, I'm going to just do a quick and dirty graph to take a look at what this looks like here. Pick the rows, insert a graph. In general, you should just do this by hand. There's no reason to do this up here because it's actually harder to actually figure out the, when you're doing the slopes and stuff. So here I've got this thing. I'm going to, again, always make sure I have titles here. This is going to be time in seconds. Trying to model best practices for y'all. So, but he didn't put an axis. So mils of oxygen. So here, this thing, I take a look at it. And what I notice is kind of what I've been talking about. Once I'm past 40 seconds or so, it's already starting to die. So if I am going to, let's go to my annotator again. By the time I'm around here, I don't care about that at all. In fact, in all of our cases, from 60 seconds on, it's really flattening out. All of the really interesting stuff where we're actually going to be able to get a good measure of the reaction rate is going to be in the first, probably 40, 50 seconds. Again, we're going to throw out the very beginning because we just saw that that is not trustworthy. We're going to throw out this part because this part is where things are dying. So let me use, I'll change my color to a red for like throw out. I'm going to throw this out because the reaction's dying. I'm going to throw that out because I have that artifact of that huge bubble coming up. which then gives me kind of my, I'll use gold, my gold zone. The place where I really care is where was the reaction really going strongly before it died, but after it had a chance to mix and get rid of that big bubble. So this is where I'm going to draw my best fit line. And maybe I'll draw that in, I use like, I am going to have trouble actually, because I don't have, let me see if I can do this. I'm trying, you can't see me, but I actually have like a ruler on my desk with a mouse, and I am trying to be super clever here. This is good enough. Oh, no, that's stuck. Oh, that's good. There is my best fit line. Use a ruler. So few things. Again, since you're using these graphs to actually get quantitative information, make sure your graph is large enough that you can actually plot things well and you can actually look and measure things well. Make sure you use a ruler when you're drawing your lines, because nobody draws a perfect line. Like, it's always ridiculous to me when people turn things in and there's this wiggly little line, and it's like, use a ruler. Then you need to do the analysis. Again, you pick any 2 points. Again, I am going to end up, let's pick 30 and 20. So what I do now is I'm going to draw, let's get, I'll use green. So at 30, ... it's right here. Notice that it's not on necessarily a data point. It's the intersection of my best fit line and my axis here. And again, at 20, I go up and it's right here. Notice it's also not exactly on the data point. So your points to calculate the slope do not have to be actual data points. It's the line, it's not the data points. That's important. And then I know my delta X here is just going to be 10 seconds. Right? I did that by design. I just picked 2 points that were 10 seconds away from each other. And then over here, in fact, I'm going to, if ideally I would have more resolution here, but let's say this is about 12. Let's say what, I don't know, let's say this is like 18.5. So what would be the slope here?
$6.5.
Over 10.
Over, yeah, exactly, 6.5 over 10. So we do rise, so our delta y, which is going to be 18.5 minus 12 over the run, delta x, which is going to be 30 minus 20. And then 18.5 minus 12 is 6.5. 30 minus 20 is 10. So my slope is going to be 0.65 moles of oxygen per second. And this number then would be the number I would use on my main graph that I really care about, which is reaction rate versus substrate concentration. But this is how I calculated. This is trial 3. So this is, would be for trial 3, the one that has 3 mils of hydrogen peroxide to 2 mils of water, or like, overall 60% of our main solution.
When you say that we should hand draw them, do you mean like the best fit line or the graphs?
I mean, definitely the best fit line you're going to hand draw. But even the graph, I find it's just easier to just draw it yourself. If you do it this way, you are going to definitely need to not just do what I did here. You would want to you would have to like
0, 1, 2, 3, 4, 5, 6.
wait a second. Make sure you have much finer resolution so you can kind of tell where things are at when you move over. And if you did it on this, you still need to make sure you are being really good with your numbering and everything. So, when I do this kind of stuff, I just do, plot it out, because that way you don't have to print it. But if you want to print it, you still need to use a ruler and a pen and draw on this, and then go extend your lines to here and make sure you are good at figuring out. Don't just like, yeah, it's around 20. You really want to be close to what it really is. You can even do minor, yeah. So anyway.
So when you're drawing the line and you're trying to find the slope, do you, how you just pick like the 10 seconds and all that, you're not doing the whole line, you're just doing a part of the line?
So you mean for calculating the slope?
Yeah.
Right,
Like
so it doesn't matter. Any 2 points that you get on a line will give you the exact same slope.
Okay.
There's no, in general, like, I can, let me give you a, do I have, let me see if there's a, oh, I don't. Okay, I am gonna, let me try to draw, oh, here it is. Okay, so let's say this is my line. Oh, I wish I saw this before. Holy crap. Okay, so I've got my little line here. If I pick points that are too close, then there's much more of a chance of like kind of error based on exactly what is this versus this and this and this. If I pick points that are kind of further apart, then any small changes in measurement that I'm getting and interpolating or whatever is going to cause less of an overall error in the actual ratio, the slope, right? If I try to measure my slope from this point and this point, these have to be really accurate. But if I get my slope from this point and this point, it's going to be the same number, and theoretically it should be exactly no difference. But the reality is I'm going to be a little sloppier in exactly what the values are, and it's not going to make a difference in the actual slope number, if that makes sense. So theoretically, I could pick my slope there, or slope here, or slope here, or wherever. It should always be the same number. But in general, you want to pick ones that are going to make your life kind of easy and spread out enough that they're not too cramped. That would be what I would say. And like I said, again, it does not have to actually incorporate any of the actual dots, right? I can get the slope up here even after it's launched away from the actual data, but the slope here is still the same as the slope here. It would be the same as the slope here. The slope is always the same thing, no matter where you look. So any questions about that? Okay.
I'm just a bit confused with the trials on the graph to know which trials 1 and 2 and 3. That's my only confusion right now.
If you go look on the bottom,
Oh, is it there in the bottom?
yeah, you click on the tab here, whichever one you click on. This is clicked on trial 3, and we have trial 3 there. But if I click on trial 2, it's bringing up the trial 2 data. If I click on trial 5, it's bringing up the trial 5 data. So
Okay.
each of the different trials is in its own tab there.
Okay, I see. Thank you.
Analyzing this lab, it's a little tedious. You're going to have to go through each. There's 6 trials. You need to get a reaction rate for each of them. Then you can use that to finally make the graph you really care about and address your actual objective and hypothesis. And like I said, people tend to get confused on this one. I think because, the graphs often look similar even when they're looking at different things. If you are confused or you're not sure you're doing it right, check in with me. I'll do my best to try to give you feedback. The turnaround. I know some people I've gotten back to, some people I still have some feedback I'm still waiting to give you. But in general, I will do my best to get back to you on things. And if you do get confused, be advised that this one tends to confuse people. So you will not be unique. In fact, you'll be pretty typical. What else? Are there any other questions about this lab?
Do the graphs also need to be in pen, or can we use like colored pencils or anything like that to show the different
Yeah,
lines and
so
everything?
in fact, okay, so that's a good point. Colored pencils, this is one of the few times when colored pencils I think is pretty good, because let me show another thing I can show you that would be interesting. It's nice to do each one on its own, but it's also kind of nice sometimes to show them all on the same graph. I'm going to look at time in seconds versus moles of oxygen. Sometimes what people do, trial 1, this was trial 6, which was 0, right? It kind of went up and then it just stayed flat. Trial 1 kind of goes up and then it kind of dies. Trial 2 goes up and then it dies. Trial 3 Your trial 4, you can, actually this wouldn't be. So it's kind of nice when you put them on the same graph, there's a few things that show up that you might not notice otherwise. So on this graph, how could I get a sense of what the reaction rates for the different trials is? Where would I find the reaction rates for the trials?
Like between the 5 seconds and, is that what you're asking, or the best fit line?
Would be a best fit line, right? If I wanted to get the reaction rate for trial 1, I would have to draw my best fit line here and calculate the slope. Right, that is what the reaction rate is for trial 1. If I want to get the reaction rate for trial 2, what would I do?
The same thing, but on the green one.
Exactly. Oops, let me do this here. And I would do a delta y over delta x for that. And what do you notice about the slope, the green slope versus the red slope?
The green slope is a lot steeper.
Yeah, the green slope is steeper, which means what?
It has a faster reaction rate.
It's got a faster reaction rate. So looking at it, comparing the trials in this way, we know that trial 2 is faster than trial 1 because the slope is steeper, right? If you calculated the number of it, it'd be a bigger number. When we go to trial 3, it's a little steeper yet. But when we go to trial 4, what do we notice? Whoops. What do we notice about this, about trial 4 and trial 3 in terms of their slope?
Pretty much the same.
They're pretty much the same, right? They're kind of parallel. What does that mean?
That the reaction is about the same.
But the reaction rate is about the same. And what would that tell us in the bigger picture?
Trial 3 is the saturation point.
Yeah, we're at saturation, right? And if we take a look, trial 3, we make a certain amount of oxygen before it's over. Trial 4, even though it was at the same rate, the same slope, it went on longer. And by the time we were done, we had more oxygen because we broke apart more hydrogen peroxide. But the rate that it was happening was the same. What if I was going to put trial 5 on here with even more hydrogen peroxide? What would happen?
Similar reaction rate, but over a longer time.
Yeah, it would have the same slope again, but then keep going up higher and continue on to make even more oxygen before you're done. But once we've hit saturation, no matter how much more concentrated the substrate is, the rate is the same, which means the slope is parallel. So this is another way to think about what saturation looks like. If the slope is getting steeper, we know that we are getting faster. But if the slopes stay parallel, then we know that we are staying at the same maxed out reaction rate. Right, so sometimes people will, and if you are going to put them all on the same graph like this and put your slopes on there, make sure you are very clear about colors, like which color is which trial. Make sure that you make it big, so the stuff that you're interested in fills up a page, so you can actually get good numbers and everything isn't all crunched and you're kind of making up your numbers. So, but yeah, no, this can be pretty if you like put them all on one big graph the size of a page and draw in the best fit lines. It gives you a kind of bird's eye view of how they all relate to each other that you don't get from the other pictures.
Would it be more useful to do like the whole data set for this graph, or just the like 10 through 50 seconds that?
Wait, hold on. Your kind of volume was low. Say that one more time.
Would it be useful to use the entire data set for this graph, or just that like window of the 10 to like 50 seconds?
You know, I would say the important stuff that to draw your graphs, draw your best fit lines would be, I would say the first 60 seconds is all you'd really need to put on here. Because otherwise you would have like half the graph, which would be stuff that is not relevant to your best fit lines. So I would say it probably makes sense to kind of magnify things so your x-axis, you know, ends around 60. If you want, you can go further on, but it's not going to be useful for your calculations of the reaction rates and the slope. It would be pretty in terms of looking at what was the ultimate amount of oxygen that got made. So, but, you know, if I was doing this like purely on a practical level of getting the numbers, I would probably just stop around 60. But yeah, so that's a good question. Any other questions about all of this stuff?
As far as the best fit line goes, how do you go about, you know, finding where to intersect with the x-axis?
It's irrelevant. Don't think about the x-axis. Just think about the points where, again, if we kind of go back, I'm going to go back to this one here. And what I basically do is I tell myself, all right, this part, ignore. This part, ignore. This is going to be the place where I care. And let us, you know, get a best fit line that is going to best approximate those. So don't even, it doesn't matter. The x-axis is irrelevant because the x-axis is actually probably going to be displaced because of that big bubble. You're not going to, you know, the reality, if we kind of extrapolate the thing back, it's probably going to cross at 5 rather than, actually, yeah, no, it's not exactly true either. I would say just forget about the x or y axes and just think about the dots that you care about and the line that best fits on them. And then once you've got that line there, then you just go and do a, you know, delta y over delta x and calculate that and use that as your rate for this trial.
Understood. Thank you.
Sure. What else? Any other comments, questions on this? If I keep looking at my background and I go, yep.